well, it's great to be there, except that I'm not there. I'm back here at the Salk Institute, but what a wonderful group and set of talks. I'm going to, first of all, share my slide, specifically here. And that is my first slide here. Okay, so I'm going to be talking about neuromodulation. And I'm not sure if everybody can see the slide. Can you see it?
Yes.
Okay. Yeah, so I hear an echo. Okay, so here's the story behind this research. So I was organizing a similar meeting here in La Jolla. This is about 2 or 3 years ago now. And we brought together neuroscientists with people in machine learning and deep learning. And it was a great talk, I mean, a set of talks, but, you know, it was clear that there was a divide. There was a, you know, the questions often, you know, were that one group would ask would not really register with the other group. Now, one of the questions that was asked by Kay Tai, who's a colleague of mine here at the Salk Institute, studies social cognition in mice, was, what about neuromodulators? Do deep learning people use neuromodulators? And there was a blank, you know, response, in the sense that they weren't even sure what we meant by neuromodulation. So that started a collaboration with Kay. And so we thought we'd explore that. And we brought in Hava Siegelman, who's also was at the meeting. Now, what's interesting about neuromodulation is that unlike point-to-point network models, the architectures that you've seen, which typically involve fast synapses, you know, excitatory inhibitory interactions, neuromodulators have a longer time scale, and they're also more global. They're diffusely distributed. And so in this levels diagram, you know, it really spans the whole, you know, stretch from molecules up to the entire, into behavior, has an enormous impact on behavior. And so it's a different way of thinking about how to integrate, you know, state changes basically into networks. Now, the history of neuromodulation is an interesting one. So, you know, people in the audience probably have heard the story about Otto Loewi, who discovered the first neurotransmitter, acetylcholine, was in the heart. But in fact, it was also the first neuromodulator, because acetylcholine, when it's released, slows the heart through a muscarinic second messenger system. And in fact, that's allowed him to isolate it through the perfusate. Now, there's a coincidence here, because when I was a postdoc, I worked on the muscarinic system in a model system with a bullfrog sympathetic ganglion cell that you see here on the right, with Steve Kuffler. He was my postdoc advisor. It was a transition from theoretical physics to neuroscience. Now, it turns out that this cell, in addition to, you know, releasing acetylcholine and having both the fast excitatory and the slow muscarinic response, also has a peptide that's released when you have long, slow activity patterns, which turns out to be a mammalian hormone, LHRH, luteinizing hormone releasing hormone. And Steve and I worked out the synaptic physiology. We bulge clamped this neuron as suitable for that because it's, as you can see, it doesn't have dendrites. And what's remarkable about this, the peptidergic response, is that it can last for minutes. In fact, it takes a minute just to reach peak, and then it can continue on, but you have to wait 10 minutes in order for it to be returned to baseline. So that this slows down collecting data, but it also has the advantage that you can stimulate, and as it begins to slowly rise, you can go out and get a cup of coffee and come back, and it's still not up to the peak yet. So in any case, this was a paper that we published if anyone's interested. I think it's the best characterized peptidergic synapse. Now, Steve and I would go out for long walks. We happened to be passing this church. And he said, "Oh, Terry, you know, Otto Livy is buried in the cemetery." And I was dumbfounded. And he said, "Sure, let me show you." So here's Otto Livy's grave in the Woods Hole Cemetery, where we worked in the summers. And at 1873, 1961. And so it turns out that he would come in the summers too. And he loved Woods Hole. And this is something that part of history. Now, if you look at what we know about neuromodulators, probably the best studied system and our understanding is really owed to Eve Marder, who studied the stomatic gastric ganglion in the lobster and crayfish for many years. And what you found is that there are, first of all, multiple neuromodulators. There's dozens, and that they act in different ways, primarily on the synaptic strengths and the intrinsic properties of individual neurons, and they can shift the activity pattern between two different bursting patterns, two different rhythms, the pyloric and gastric mill rhythms that differ from each other by an order of magnitude in period, and completely reconfigure the whole network. It's really remarkable that you can get two different networks into the same physical connectivity pattern. And again, what this shows is that you need to know the physiology if you want to understand the dynamics. Just having the connectivity pattern is not enough, and it's especially true in small ganglia like this, but it's also true, as you'll see, in the mammalian brain. So here are three classic neuromodulators. Dopamine, you've probably heard about that already in terms of reward prediction error, but also noradrenaline and serotonin. Now, acetylcholine has a very interesting impact, again, through the muscarinic, central muscarinic receptors. It regulates through the presynaptic terminals, receptors on the presynaptic, it regulates the strengths of synapses in the hippocampus. And Mike Hasselmode studied this several decades ago. And what he showed was that depending on the state of the animal, if it was exploring, then the acetylcholine would reduce the strengths of all the recurrent connections, the strengths of the recurrent pathways in the CA3 area of the hippocampus, which is very densely innervated with recurrent connections between the pyramidal cells. But that when the rat stopped and began introspecting or eating, it would, whisker, grooming and so forth, those strengths would be upregulated, and now it would become autonomous and would actually be activated and produce patterns of activity, these so-called ripples that would go back to the cortex. So this shows that you can reconfigure even in the mammalian brain through the similar mechanisms. So here's the project that I'm going to tell you about, and this was work that was done by Ben Suda, who's an MD PhD student here at UCSD. And we used deep learning methods to train recurrent networks in order to do a simple task, and it's illustrated here in the upper left. Bears love mushrooms. I didn't know that, but it's true. Now here's a bear who has encountered a new mushroom, and the bear knows that there are some mushrooms that make you sick. And so the question is, should I approach it and try and eat it, or should I avoid it? And this is modulated by the level of hunger. If you're not very hungry, you just pass it by, and if you're really hungry, you might give it a try. Okay, so this is a classic go no go task, and it's illustrated here in B, where there's going to be two different inputs. That is to say, either there's no input, or there is going to be on the same input lines into the recurrent network. There's going to be a stimulus. Now, this recurrent network has a few hundred units in it, 20% inhibitory in red and 80% excitatory. So we have a little bit of a constraint, architectural constraint, but it's really a simple recurrent network and trained with backprop. And here's the traditional go, no go is that if there's no input, there's no output. But if there is a stimulus, then you have an output, which then says that the action should be taken. Now, here's what we did. Here's our twist on this very simple behavior. What if we decrease the strengths of all the synapses by 50% and trained up the network to produce the opposite behavior when those strengths are reduced? In other words, in this case, the no input should give you a reduction in activity in the output, and the stimulus should produce no output. And the question is, can you train the network to do both under the two conditions, one with neuromodulators and one without? So here this, you can see the weight distributions, it's compressed when you add neuromodulators. And the answer is yes, you can train it up. It does very well. And here's the traditional go, no go of the network producing a very nice output for the input stimulus. And here's in the neuromodulator condition, it does just the opposite. And the population activity is different. And we can look at individual neurons. And the question is, what's happened to the patterns of activity? Well, some neurons, as you can see here on the left, have similar activities during the time course of the making a decision. But on the right, you can see there are other neurons that have really dramatically different time courses under these two conditions. And so there's a range, there's a broad range. Now, one of the advantages that we have here is that we can go into the network and we can try to understand the logic. In other words, what's happening in the interactions between the units because of the fact that we have added a neuromodulator. And we've trained it up to do two different things. And in this, by the way, we train up many networks and they could all produce the same output, although they're different. And this is one of them that I'm showing you just as an example. But by clustering activity, we can see that there's 5 different groups. There are 2 that are inhibitory, 2 and 5. And then there are 3 that are excitatory. Now, each of the vertical lines is 1 neuron, and the color is the average firing rate across the entire task. And so you can see the ones that are in yellow are the ones that are highly active, and the ones that are dark are the ones that are not active on average. And focus first on the 2 inhibitory clusters. You can see that they tend to be highly active only in certain for certain types of stimuli and neuromodulatory conditions. So they're clearly important for signaling what the state of the network is going to be and controlling the output. And that's true for 2, less so for 5. And you can also see that there's a complementarity between 3, which is excitatory cluster, and 2, which is inhibitory. So there is a logic that's formed between these different clusters that produces this behavioral output, which can be shifted. If we vary the neuromodulation level, we can see that the output is going to be modulated, and each one of these is a different network. And so you can see that they don't converge to the same network. There's a lot of different networks that produce the same output for high modulation levels, even though, as you can see, at intermediate levels, they may have different behaviors. And we never regulated the intermediate behaviors, so it's not too surprising. So we then launched into a number of different versions to test to what extent we could reduce the number of modulated units. And there's evidence now that for many neuromodulatory systems, it's not completely global. It's not the case that one neuron projects to the entire cortex. It may be only to a certain patch. So the first thing we tried was to see what would happen if we just modulated either the excitatory units, shown here circled with blue, or the inhibitory units in terms of changing the synaptic strengths, decreasing them by 50%. And the answer is that the test performance is the same, whether you do the excitatory or the inhibitory. It takes a little bit more training for the excitatory modulation. What about three behaviors? What if we increase the strengths by 50% and decrease by 50%? Can we get three different behaviors? And so here's the two behaviors we've already shown. We added a third behavior where one goes down and the other one goes up. And as you can see, there's no trouble. It learned it extremely well. And so we can actually get three different behaviors into the same small network. Now, how big of a, how many neurons do you need? What if we vary that from 0% to 100%? And the question is, what's the performance and how long does it take to train? And as you can see here, it's pretty much identical in terms of the performance, even going down to 10%. So you don't have to modulate the whole network. And it takes roughly the same number of training trials in order to achieve that performance. And so we tried to push it to the limit. Okay, 10%. Well, let's see if we could reduce increase the number to 9 different combinations of outputs. And could you hold on for a second here? Beatrice, I'm on Zoom. And it was fine. It could do 9. We didn't push it beyond that, but it's pretty clear that there's a lot of flexibility here. So we wanted to actually see whether we can reproduce some experimental data. And so here's experiments that were done on Drosophila. And here's the experimental data here at the top, which is the Drosophila, as you can see, they varied the concentration of sugar, and the question is, what concentration do you need before the fly actually starts lowering its proboscis, the proboscis extension response? And if it's sugar fed, you can see you have to go up to very high concentrations, 100 millimolar. But if you water deprive the fly, it will start licking at very low levels. One day, the threshold here is 25, went down by a factor of 4. And then for 2 days, it really is thirsty. And so it'll start with very low levels. And then here are the averages over many flies. So here's what we did. We trained up a recurrent network with just 10% of the weights in the network changing strength. Under the 2 conditions, one where we mimicked, as you can see here, the condition where it had been sugar fed, and we actually trained it on this curve here, and we also trained it on the case where it was water deprived. And then we varied the degree of modulation, the strength of the synapse between the synapses between the 2 limits, and we got a very graded response. And so we could reproduce the whole spectrum here. Presumably, if you've changed the amount of time water deprived, you could fill it in here, and we could reproduce that. And so what this shows is that this is very flexible, it's very adaptive, and it can explain phenomena like this. Now we can go into the network and look at the trajectories. We do a principal component analysis, and here are the 3 top components. And here is the trajectory starting at t equals 0 for 2 different curves for the neuromodulator either present, which is in blue, as shown here, at different stages, and then when it is absent. And you can see that the trajectory is going off in different directions and produces different outputs. And if we change the neuromodulation factor, we can fill in the trajectory varies between these 2 extremes. So we see that the dynamics itself is something that can be continuously moved around in the space. And this is something now that has been studied very great detail by several labs, including David Tank, in an experiment where the mouse had to make a choice to go right or left. And he showed there that you could very nicely show that there's a subspace, the trajectory goes through the subspace, and that the actual path that it takes is within the subspace actually varies. So it's not, and you can see here, in fact, that we've averaged across tasks here, and the width here, it gives you a sense for the variability in the activity patterns. And that's what David Tank has observed. Now, EI balance has been a important metric or a important measurement that has proven to be important in understanding activity patterns, the balance of activity in the cortex and other areas. And so we wanted to see how that variable, we looked at the ratio of the total excitatory strengths to the inhibitory strengths, the ratio. And indeed, that varies. Each is a particular network. And so it's a range of these ratios. And you can see that the proboscis extension here is related to that ratio. And then we wanted to parse it down to subsets of activity patterns of strengths. And we broke the network, the weights down into either the, in number one, which is the modulated recurrent network weights. Number three is the weights within the non-modulated group. And then the connections between those two groups, 2 and 4. And as you can see, that this when we looked at this, the correlation coefficient here, the only one that actually had any impact was the 3, which is interesting. So it turns out that it wasn't the weights within the modulated group that were controlling this relationship, this correlation. It was the ones that were in the non-modulated group. So there's an interesting, there's a lot more work to be done here, but it's pretty clear that we can extract some principles out in terms of how you embed multiple tasks into the same network. So here's a cartoon of what we think may be happening. So the idea is that if you train up a single network, but 2 networks on 2 tasks, you come up with 2 different sets of weights. This is weight space. And here's task 1 and here's task 2. And obviously there's a gulf between them because they have 2 different outputs for the same input. But when we modulate the strengths, we can see here that we can move from the T1 to the modulated T1 and then move it over so that it is able in the modulated state to adopt the behavior for the T2 weights. Okay, well, that's the story. There's a lot more that we've done, but running out of time. I want to tell you to end with looking into the future. Where is this headed, both in terms of the artificial neural network business, deep learning, but also what implications does it have for neuroscience? And I think that we've reached a point now where for the first time, artificial intelligence and neuroscience are on the same page. That's say we're using similar vocabulary. We're talking about units and neurons. We're talking about synapses and connection weights. We use different words, and it's a vastly simplified version when we looked at the deep learning networks. But at least the mathematics might be similar, and as we've shown and many others have shown, we can learn. We can learn about how different architectures and different constraints put on the architecture can affect the performance. And Delia's talk, I think, showed that it can give insights into some of the things that we know from psychophysics, and now we can pinpoint into the network specific mechanisms underlying both the recordings and also the performance in psychophysical tasks. So this is a paper in PNAS that was published a few years ago, and it came out of a conference, a symposium that was organized by the National Academy of Sciences, and it was organized by mathematicians, not by neuroscientists or even engineers, by card-carrying mathematicians and statisticians. And the title of the meeting was "The Science of Deep Learning." And the audience actually were field medal winners and some very distinguished mathematicians. And the upshot of the meeting was that the mathematicians are puzzled. And the reason they're puzzled is there are paradoxes that the performance of these networks, both the training and the ultimate performance, according to classical statistics, sample complexity and optimization, non-convex optimization, it shouldn't work. And it does. I mean, that's empirical, right? Simulations. And this is an opportunity. When you see something that is a paradox, this is an opportunity for maybe discovering new mathematics. And that really was the theme in the meeting. And that was the theme in this particular essay, The Unreasonable Effectiveness of Deep Learning in Artificial Intelligence. And let me cut to the chase. Here's where I think, and many of the others at the meeting thought, we're going to really make discoveries. If you look at the history of the fields, optimization and in statistics, by and large, people worked with small models and they were trying to find a unique set of parameters. And what they found is you had too many parameters, you overfit, and it would not be able to generalize. And they proved theorems. Well, that's true for low dimensional spaces, but it turns out not to be true for high dimensional spaces. And now we're dealing with spaces with millions of dimensions, in fact, billions and billions. And it keeps getting, they keep getting bigger and more layers and more complexity, tension, and all sorts of other mechanisms have been put in, and performance gets better and better. And what's interesting about this is that it's a whole, once you get into this new regime, over-parameterized regime, then it really, everything changes, and you really have to reanalyze it. And I'll just give you one insight that came from the meeting, which is the following, that if you do convex optimization, you're looking for the unique set of parameters that optimizes a particular network or task model. But if you have over-parameterized networks, vastly over-parameterized, then it turns out for every starting point, you reach a different out network at the very end, where you do have a minimum. But it's and they're all different. There's an infinite number of networks that have roughly the same performance, right? So you've changed the task from finding a needle in a haystack, which is the problem that statisticians solved very, very elegantly in the past, to finding a needle in a haystack of needles. And I think this is where biology is. You can have two people who learn the same task, and very likely their connections in the brain and how they solve the task is going to be quite different. There's many ways of many networks that produce similar behaviors. And I started this little essay with one of my favorite books called Flatland. It was published in the 19th century by Edwin Abbott. And it was actually a parody of Victorian society. And in this society, in this particular flatland, this two-dimensional world, all the creatures were, as you can see, squares and triangles. They're geometric shapes. And the more sides you have, the higher your status in society. And of course, the king was a circle. Now, the square, the gentleman square, has a dream one day where he imagined that, in fact, the circle is actually a cross section of, there's another dimension, and that there's this dimension where the circle is a cross section of the third dimension passing through it. And he got all excited about this and went around talking to people, and everyone thought he was crazy, and he was eventually incarcerated, right? Now, this is just going from 2 to 3 dimensions. And, I think we're living in the equivalent of a flatland, because, our brain grows up in a 3-dimensional spatial structure. Of course, there's time, but the reality is it's very difficult for us to imagine what the 4th dimension would look like and how it interacts with our dimension. A few people can do that, but, but let alone 1 million dimensions. That the type of geometry and topologies that are possible out there are enormously complex. And this is where I think there's going to be progress in the future. In terms of mathematicians helping us understand why these networks work, under what conditions they work, and to help improve them. So finally, this is the team, Ben, as you can see, and more recently an intern, Stefan Pate, and then Kay and Hava, who have been great colleagues. So I'm sorry if I've gone over, but in any case, hope that there's time for some questions.